Well, hello, and welcome to Bio 121. And what we're going to be covering today is how do actual cells communicate with one another. We know that between two human beings, actually having a complete conversation where everybody walks away with an understanding of what the intent of the conversation was and how it was executed, and then how it was interpreted, oftentimes is actually really difficult. And when I'm saying that, I'm thinking to myself, how is it I put together, presentations or lectures or these things here, to make sure that you understand what needs to be communicated so you can understand how to use the information afterwards. And inside Chapter 9, some of the really good things that are done inside of it, and I'm hoping you walk away with an understanding of, is how it is that we can go from a concept of what signaling is and using experimental data, and I'll try to explain some of it to you as we're going along, how Mother Nature created a series of systems that allow for nuanced communication. I don't mean nuanced as in like, 3 droplets of this means something. What I mean is Mother Nature needs to create specific sets of signals such that unique events occur inside of a cell. So if we think about, let's draw a cell for you here, right? Here's a cell. Draw a cell, right? Here's one cell here. Let's put another cell over here. And one cell is going to say, hi. Or in computer parlance, hello world. Okay? And somehow that signal can either be collected at the cell membrane, if you think about that as inside your ears collecting information from the outside world. Something will bind this and cause a series of events that are going to occur inside here. And this will then respond. Respond. Okay, but you don't know what the response is until you watch it for some period of time. And sometimes there might be an instantaneous response. INSTANT. Or at least it appears as though it's instantaneous. Okay. Others can be delayed. DELAYED. Okay, and delayed for, 5 minutes. Could be delayed for 8 hours. And the point is this, is when we're thinking about what communication means, inside Chapter 9, we're identifying those repeated observations that have been seen in the world. And now we want you to leave this chapter with an understanding of what Mother Nature is trying to accomplish. Okay? And that's a really important idea, because if this is a one-sided communication, then maybe this means that this is the way the entire signaling system has to work. But more often than not, if we think back to Chapters 1 and Chapters 2, there's a feedback system usually in place where some information is sent to one place, some control center is going to interpret it, and then some effector will then allow for a response to occur. And the production of that effector, much like we saw in Chapters 6, 7, and even part of 8, will then signal back here so that we then adapt our responses to what's actually going on. Sorry, I blew that up just to make sure you can see. This would be some set of receptors that send information to a control center, and then the effector here, the production of the effector could, in all likelihood, literally change the entire signaling system. Okay. So having said that, let's move on. There are 4 categories of signaling when we think about what we see inside the natural world. Paracrine signaling, endocrine signaling, autocrine signaling, and direct signaling through those gap junctions. Remember, those are made of those connexin proteins, and they allow materials to move, and they can move in 2 directions here. Each of these require a molecule to be produced inside of a cell, and then, excuse me, and then exocytosed, and then it's going to do its job. So that means our first cell, if we go think back to the previous image, our first cell has to know exactly what it needs to communicate and how it's going to communicate that. When we think about the difficulties we have with, trying to say hello to somebody when you don't know, your brain becomes flooded with all these different possibilities and scenarios. But the reality is all you really need to do is go over and say hi. Okay? But And what that actually means in biology is something that looks like this. Autocrine signaling systems are cells that make their own signal, they have the receptor for that signal on the cell, and that's what regulates autocrine signaling. Signaling through gap junctions, through those desmosomes, through those connexins, they allow ions to pass between cells. Sometimes they can go in the other direction, but the idea is this, is the signal doesn't actually have to leave outside of a cell. You don't need to use the mechanism of exocytosis. The presence of the channel here will allow it to pass just a little bit more rapidly. And then these two are more synchronous in their responses. So if the signaling starts here, this cell will know what's going to happen. Paracrine and endocrine signaling systems both use exocytosis. The target of a paracrine signaling cell, though, could be to travel through a specific distance or space and then bind to the receptors that are on that target cell. Each of these orange dots has a unique responsibility. It's not as general as the cartoon is suggesting for you here. And what you want to pay attention to, of course, is that when we actually start thinking about how this might work, Mother Nature can then become a little more, precise. She can have a specific type of cell that will make a specific molecule and exocytose it into the circulatory system. And then it will travel throughout the entirety of the body. And in doing so, any cell that has a receptor to that molecule then becomes the target cell. And that target cell then will respond through a series of metabolic responses to that now new signal. An example of paracrine signaling systems can be seen here, where we have a presynaptic neuronal cell and a postsynaptic neuronal cell. Big fancy words for it. Let me draw this for you. Okay, I'm going to draw one multipolar neuron for you here. Yay. And this is a multipolar neuron because here are the dendrites in the cell body. Here's the axon of that neuron, and here are the axon terminals. And what they've drawn for you here is they have another cell over here. Okay, let's go like this, go like this. And what they're doing is, I don't know what color are they using as a neurotransmitter. They're using green. Let's go to green here, right? Let's go green. What they're doing is they're saying, look, this neuron here is releasing this molecule here. And this cell here has the receptors to that molecule there. And what's going to happen now is that some signaling mechanism is going to occur on the inside of this cell as a result of the signal that's occurred at the surface. This is what we mean by paracrine. Diffusion is the major mechanism, and the diffusion distance is usually what causes the timing inside of this system as well. And that's very important. You don't want synchronous activities inside some parts of your nervous system. So when we're thinking about what actually is going to happen next, internal receptors are an example of something we've seen before, where hydrophobic signaling molecules can literally pass through the membrane. Remember the general rule, like dissolves like. So let's go to yellow here. And I'm going to say that, look, oops, you can't really see that, can you? Let's try something else, Jonathan. Let's go with this. Maybe this will work better. In fact, it does. Okay, 1, 2, 3, 4. And remember, this is a 4 cyclical carbon structure. We consider these to be the basic building blocks or cholesterol is the basic building block for hydrophobic signaling molecules. And remember, from here, we built the steroid hormones, steroid, S-E-R-O-I-D, and the steroid hormones, because they're lipid, they can pass through the membrane. Literally, they just diffuse. And what happens is the receptors now inside the cell, so the molecule passes through that membrane there, binds to the receptor inside the cytosol of the cell, and will now travel to the nucleus, where it will now regulate what's going to happen here inside of the cell. So these internal receptors, generally speaking, are hydrophobic signaling molecule specific, and they tend to regulate things like gene expression, which we come to in a few chapters a little more rapidly. Having told you that, get rid of this here. Let's go here. There are many types of cell surface receptors, right? So One, we can have ion channel linked, we can have G protein, we can have enzyme linked. And what I'm going to do is I'm going to pause here. Actually, no, we can do this. What we're going to do is we're going to go through examples from each of these. And the first example is going to be the ion channel receptor, where some signaling molecule here in yellow binds to the binding site here on a receptor. And what's going to happen is because this has bound that receptor, it changes its shape, and these little purple dots now are allowed to move in. We're going to call these purple dots, for the purpose of this example, an ion. Remember ions: sodium, potassium, chloride, calcium. Each of these dissolved molecules, okay, is a charged entity, which means it can't pass through that phospholipid bilayer. So when the pore opens, that allows the ion to move inward. You're like, yay, what's the big deal? Well, we've now changed the resting membrane potential at that membrane. So what Mother Nature can do is she can use charge differences on these membranes to actually figure out what that cell might need to do next. G protein linked coupled receptors are something a little bit different. These are what we generally call metabotropic receptors. And these metabotropic receptors, they're at rest. You can see they form a complex structure here where the outside facing protein can be regulated, and then the internal structures, the cytosol inhabited structures, will now move on to do their job. So when we become activated, a different signaling molecule will bind here, and we will displace this heterotrimeric complex. Big fancy words, but just saying 3 proteins that work together. What happens now is another molecule moves into this space, and what's going to happen is Mother Nature will dissociate all of these signaling steps. And for it to actually do its job, we are going to hydrolyze that molecule. So what's going to happen is we have become GDP, and we'll now continue to move through this. So it's a cycle, but notice where it started was out here. The signaling molecule had to bind to the G protein coupled receptor, displace now this complex temporarily, allowing for a signal to move into the cell, and we'll come back to what that cellular response may be later. Now, enzyme-linked receptors are an example here. Your textbook talks primarily about tyrosine residues being phosphorylated on the inside. And I don't know what that means to you guys, but tyrosine is one of those amino acids, and it can become phosphorylated. And what will happen is, at rest, the receptor tyrosine kinase here does not have a ligand attached to it. What will happen is, once the ligand binds the molecule, you'll have a series of cross-phosphorylation events here, causing these two structures to dimerize. They're not dimerized here, they're dimerized here. Literally, they form this complex, and these phosphorylation states now determine what that residue will be. Okay? I mean, by residue, I mean, imagine, if you will, you can see a combinatorial effect occurring here. So we only phosphorylate 1 and 3, you get one set of cellular responses. If you only do 2, you get a different set of cellular responses. Or if you have no phosphorylation, that's a different response altogether. So you can see what Mother Nature can do. She can create now complex signals that may enter the cell and then have downstream effects. Here are those cholesterol molecules I tried to draw for you before, like 1, 2, 3, 4. You're like, but that looked nothing like your cartoon. Cartoon doesn't have to look like what it looks like. The cartoon has to remind you of the general structure. And we can take that cholesterol, we can build 2 additional types of molecules. These 2 here, estradiol and testosterone, higher concentrations of estradiol inside females, higher concentration of testosterone inside males, but both sexes have both of these hormones. Remember what I said earlier is that they can pass through the phospholipid bilayer and therefore drive transcription and translation. But they are not the only molecules that can bind internal receptors that are unique to those ligands. You can use thyroid hormones, you can use gases such as nitric oxide. They will behave the same because of their chemical properties that allow them to pass through the phospholipid bilayer. Now, this is a pretty good place to pause, but I want to introduce this idea, and what we'll do is we'll go backwards and we'll start all over again. What we can do is we can say that if there's a signal that's actually gotten into a cell, what's going to happen to it? Okay? In some ways, we might require some dimerization event to occur. Some ways, we're going to have some signaling event that's going to occur inside the cell. But at the end of the day, we have to have some type of integration that will allow for a response to be measured inside the cell so the cell will know what to do. So I'll pause here, and then I'll begin the next one shortly.